Okay, so hello everyone. Welcome to our today's Immune seminar. It's absolutely my pleasure to welcome Professor Sergio Quedaza from UK to give us a lecture today. So Dr. Quedaza is currently a full professor of cancer immunology and immunotherapy, also group leader at University College London Cancer Institute. So a very brief introduction for Sergio. So Sergio currently, as I mentioned, is full professor at University College London. He got his undergraduate degree in Chile, South America, and then completed his PhD at Dartmouth Medical School in US. Now, in 2004, he joined the lab of James Allison at Memorial Sloan Kettering in New York, where he investigated the machinery behind the anti-tumor activity of anti-CTLA-4 antibodies. From 2010, he started his independent research program as a group leader in immune regulation and then tumor environment immune therapy group in University College London Cancer Institute. From 2008, he was promoted as a full professor in cancer immunology and immunotherapy in UCL. So Sergio's lab is, I think a lot of us in the audience probably already quite familiar, is mainly interested in cancer immunology to study tumor microenvironment, regulatory T cells, and immune checkpoint blockade. His research team has highlighted the critical role of FC receptors and the tumor microenvironment in the mechanism of action of anti-CTLA-4 antibodies. His lab is really dig deeper into this area and then holds key patents supporting the clinical development of antibody targeting immune checkpoints, including VISTA, ICOS, and the CD25. His team co-lead the development of the best-in-class Treg depleting anti-human CD25 antibody, which was acquired by Roche in 2018 for clinical evaluation in solid tumors. Now, in addition, Sergio research also aims to characterize and examine immune activity and function within the tumor environment across different human cancers, help to identify machinery of response and resistance to immunotherapy. His work in this area really actually leads to the finding of a company called Achilles Therapeutics, a clinical stage company focused on delivering personal Treg T cell therapies in melanoma and lung cancer. With all his really remarkable achievement, he received numerous honors, which I'm not going to list. It's a really long list. Just a few highlights. Sergio got Dartmouth John Stolber's medals for excellence in biomedical research, Cancer Research Institute New Investigator Awards, Cancer Research UK Career Development Fellowship. He is also a fellow of European Academy for Cancer Science, and then most recently he was elected as a fellow of Academy of Medical Science in UK. It's really exciting to have Sergio with us today, and then we are very much looking forward to our talk.
Thank you, Wu. I really appreciate the invitation. It's really cool to be able to be here and share with you guys some of the work from my team. So I'm going to talk to you guys about regulatory T cells and cancer, and this is really quite a historical perspective of the work my lab, the work of others, and it's really a story of iterations. And I hope that's the message that I can convey to you guys, that mechanisms can lead to new therapies, and new mechanisms will help you understand how those therapies work or fail to work, and whether you need to iterate and whether you can iterate or not. So just to level the ground, there's a very low level of introduction. There are some antibody functions that are quite relevant to cancer, as you will know, right? There is the classical blocking antibodies. Two examples, you will say, the anti-CTLA-4 and the anti-PD-1. These antibodies are supposed to bind just to your target, right? And block ligand receptor or ligand target interaction. It's important that these antibodies are on an isotype in humans that is the classical IgG4 or IgG null. And the reason for that is because you don't want these antibodies, if you want pure blockade, you don't want these antibodies to engage with Fc receptors present on innate cells. Why is it that you don't want these blocking antibodies interacting with Fc receptors? Is because the other relevant activity of antibodies is antibody-dependent cell cytotoxicity or cell phagocytosis, where the antibody, for example, rituximab, binds to a target, right, and then engage with Fc receptors on myeloid cells or NK cells. And that engagement makes the macrophage either phagocytose the target that is now decorated with antibody, or the NK cell to kill the target, right? So those are two activities, and it's essential to distinguish when you want pure blockade from depletion or elimination of a cell, and that activity depends on engagement with Fc receptors, right? The difference in the activity of these antibodies, blocking versus depletion, is absolutely impacted by three key things. As I mentioned, the antibody isotype, right? So in humans, a G4 will be non-depleting usually, and a human IgG1 will be of depleting type. The type of Fc receptor that is expressed in the microenvironment, that's very important because these are not constitutively expressed, and you have activating and inhibiting Fc receptors. These are pretty much the checkpoints of the innate compartment. And the other one is target density. You might have a depleting antibody, but if you don't have enough target on the surface of the target cell, you're not going to engage and you're not going to decorate the target properly. Hence, you're not going to be able to bind to Fc receptors later on NKs and macrophages. Right. So again, in the context of immunotherapy, it will be the logic will be that anti-CTLA-4 and anti-PD-1 antibodies, because you want them to block the checkpoints, they should not engage with Fc receptors. But in reality, it's not so simple. So many years ago, when I started working on anti-CTLA-4 antibodies and the mechanism of action, what we discovered was that actually this antibody was exerting an additional activity to that of checkpoint blockade. And that activity was Treg depletion. And just summarizing many years of research, but basically regulatory T cells inside tumors, they express a large amount of CTLA-4 or much more CTLA-4 than an effector cytotoxic CD8. So then you have more CTLA-4 decorating the NK cells, sorry, the regulatory T cells. And what that does is that when then this Treg decorated with anti-CTLA-4 passes near a macrophage or an NK, that Treg is eliminated. And on top of it, you have the checkpoint activity of the antibody, which when it binds to CD8 cells, right, prevents the interaction of CTLA-4 with B7-1 and B7-2, allowing them to interact with CD28 and fully activate your effector compartment. The CD8 cells are not depleted because they express very little CTLA-4 on the surface. And that was what I was trying to tell you at the beginning, that target density is essential, is that difference between having a lot of CTLA-4 versus very little CTLA-4 that makes the tremendous difference in terms of, oh, it's acting as a checkpoint for the CD8, but it's acting as a depleter for the Tregs. But you can see that although that concept is solid in terms of data, in humans trying to apply this, it gets tricky, right? Because CTLA-4 is a molecule that can be further upregulated on cytotoxic CD8 cells, right? These systems are not fixed. So this antibody has two activities. One is depleting and the other one is checkpoint. And there's been a lot of additional data validating this, but this is us repeating this, the initial experiments we did to prove this as we were working on a collaboration with Ido Amit, trying to understand on the single cell level, what does anti-CTLA-4 antibodies do to the tumor microenvironment? So this is kind of the classical experiment that you will do to know or not whether the depleting or the Fc engaging activity of the antibody plays any role in the anti-tumor activity. So what we did here, we had mice with tumors and we treated them with controlled immunoglobulin. We had mice with tumors that were treated with the anti-CTLA-4 that everybody uses, the clone 9H10, but that we engineer to bear a mouse IgG1, right? And mouse IgG1s don't bind well to activating Fc receptors, so they cannot deplete. So in this case, what we're testing here is a pure blocker. So this will be a pure checkpoint inhibitor, anti-CTLA-4 antibody. And what you see here is that one of the mouse do delay tumor growth, but the vast majority of the mice continue growing tumors with no problem. So just blocking CTLA-4 actually at the physiological level does not lead in mouse models to tumor control. And we've done this at nausea. So in mice, depletion is essential. And here you see the antibody that has both blocking and depleting activity. And the way we do that, we use the same single chain Fv. We engineer the Fc portion of the antibody to be a mouse IgG2A, which is the classical Fc receptor engager in mice. And then in two different tumor models, we have 100% complete responses when the antibody is able to do both things, to block CTLA-4 and to deplete the Tregs that are expressing the highest levels of CTLA-4. And this type of data has been repeated by many beyond us. Now, at the single cell level, I just mentioned this because we thought this was quite interesting, and I'm already trying or describing to you that the anti-CTLA-4 antibodies originally conceived just as checkpoints actually have two activities, checkpoint and Treg depleter. When we look at the single cell analysis of mice with tumors treated with a pure checkpoint inhibitor anti-CTLA-4 or the anti-CTLA-4 that blocks CTLA-4 and depletes Tregs, we found some interesting difference on the level of the Treg. So this is the total TME. So all the T cells are on the left, all the myeloid compartment is on the right. Many different subsets. I'm not going to enter in details. So this is the density of expression of the different immune subsets. And what you see here is that this group, this is the control, and this is the anti-CTLA-4 treated mice. These are the mice treated with the anti-CTLA-4 mouse 2A, the depleting. You see, these are the Tregs. There's that population here. You give the anti-CTLA-4 that can engage with Fc receptors, and you completely lose the Tregs. So that just validates our previous data. What is very interesting is that this is the myeloid compartment and the density of different myeloid subsets, inflammatory, non-inflammatory. The anti-CTLA-4, the control animals, this is the density of those cells. When you treat the animals with anti-CTLA-4 that can engage with Fc receptors and deplete the Tregs, you see a huge change on the distribution of the myeloid compartment within the tumor. So, and we have done this experiment 12 hours post-injection of the antibody. So this is not a long-term consequence of more inflammation because you deplete Tregs. Basically, what we showed is that the engagement of Fc receptors by the anti-CTLA-4, by the Fc portion of anti-CTLA-4, leads to myeloid reprogramming. There is a huge level of activation of the myeloid compartment. And interestingly, the anti-mouse CTLA-4 that doesn't engage well with the activating Fc receptor, the mouse1 isotype, does not lead to the significant amount of changes that we observe with the antibody that can engage Fcs. And this is just quantification of that. These are all the different myeloid populations from highly inflammatory to highly suppressive described in the literature. When you treat the animals, this is 24 hours later, with the anti-CTLA-4 that is just a checkpoint inhibitor, there is no significant or not much of a significant change on the myeloid compartment. When you treat with the mouse IgG2A isotype, you have a huge increase on a set of inflammatory tumor-associated macrophages and a decrease on the inhibitory macrophages. So actually, CTLA-4 has three activities: checkpoint inhibitor on the effector cells, depletion of regulatory T cells, and can remodel also the myeloid compartment, which are described here, and those are the papers that support that. So again, for many years, we've been going deeper in this notion, also to try to develop better therapeutics. Should we develop a better, more powerful anti-CTLA-4, or are there other targets that we could take forward? And I'm just going to tell you the story of a particular target, CD25. We like working on old stuff. This is a very old target that everybody was very excited when Shimon Sakaguchi described CD25 mark regulatory T cells in mice. But then after a couple papers and some clinical data, everybody basically decided this is not a good target. Right? And I'll tell you a little bit why. So CD25 is the high affinity receptor for interleukin-2, right? It presents or transpresents IL-2 to the low affinity beta and gamma subunits, and then you have the signaling complex. Shima Sakaguchi and others described that CD25 is highly expressed on a naive animal, on the regulatory cells, and not on the effector compartment. And that made it a very nice potential target for Treg depletion. But obviously, we all know that CD25 is also used as a marker of T cell activation. Now, I will challenge that a bit, and will say, yes, it is a marker of effector activation. It gets upregulated upon effector activation, but it's context dependent. And I will show you the data why. But primarily where you see clear upregulation to very high levels of CD25 on effector cells is in vitro, when you're doing anti-CD3, anti-CD28, when you're putting IL-2 in the culture. And in vivo, when you're working with TCR transgenic models or looking at antiviral T cell responses. So there is an issue there with signal strength, I believe. Why do I say it's context dependent? I'm going to show you the context of cancer, because that's the context that I work on today. These are 3 tumor models from poorly immunogenic P16 melanoma to highly immunogenic chemical-induced MCA205 sarcoma. And this is looking at TIL, tumor-infiltrating lymphocytes, and we're looking at the surface expression of CD25. In all of them, but focus on the most immunogenic one, where the CD8s have granzyme, they're proliferating, etc., etc. You see that CD25 is primarily expressed at very high levels, target density, on the CD4 positive, FOXP3 positive, not expressed at those levels on the CD8 effector compartment. If I gate on the CD8 PD1, it's not there. On the CD8 granzyme positive, it's not there. Yes, you have expression of CD25, but it's very low, right? Again, target density. And for the helper CD4 positive, FOX negative, there is a decent, small but decent sized population of CD25 positive cells, and those are CD25 positive, PD1 positive. So maybe it's not a good idea to deplete them. Maybe it is, but we'll see. So that's mouse, right? And one could say, well, mouse is just a model. So this is human. So this is human melanoma tumors, not so small lung cancer tumor, and renal cell carcinoma tumors. And what you see again, first, that in the blood, CD25 is expressed on the circulating Tregs and not on the effectors, or in a small fraction of effectors. And in the tumor, at varying levels, it's also preferentially expressed on the FOXP3 positive cells and not on the FOXP3 negative or on the CD8 positive cytotoxic cells. So both in mice and in humans, target density, to me, looks like a pretty decent target. And although we can say these are all treatment naive patients, right? So these are primary tumors. So maybe there is not enough activation for CD25 to be upregulated. So we did a small trial in the context of a small trial set up at the Royal Marsden in collaboration with James Larkin and Samra Turakli. We were able to access patient biopsies, and this is in renal cell carcinoma, patient biopsies prior to initiation of anti-PD1 and then 6 weeks on anti-PD1. And these were same site biopsies. And what I hope you can see here is that the CD25 in brown is associated primarily with the blue cells, FOXP3, and not with the red cells, CD8s, in baseline. On therapy, on a patient responding, where you have activation of the CD8 compartment, the picture remains exactly the same. So even in the context of anti-PD1, we don't see upregulation of CD25 on the effector compartment. And this is the quantification done by the pathologist in samples from renal cell carcinoma patients, and the same type of experiment in patients with melanoma treated with anti-PD1. CD8s, either on baseline or on therapy, don't express CD25. This molecule, this target, is expressed only on the FOXP3 positive cells. So all of this to me has always been, it's a good target. Now, why was it dropped? It was dropped by this type of experiment. Many years ago, I can say, Jesus, 20 years ago almost, we and many others were very excited about the anti-CD25 antibody and what Shimon had described. The clone that I'm sure that all of you, any of you that have ever tried to deplete a Treg for any of your experiments, be cancer or autoimmunity, you use clone PC61. And quite important, the isotype of clone PC61 is RAD IgG1. The experiment that we all did, right, initially was, because we wanted to do this properly. You take your favorite therapy. So I was in Jim's lab at this time. My favorite therapy, a vaccine plus anti-CTLA-4. Jim and many others, and we showed that if you have a mouse with a melanoma tumor, you give that therapeutic combo 3 or 4 days post-tumor implantation, the tumors get rejected, right? But to test Treg depletion, you take that favorite therapy and you make it suboptimal. So instead of treating the mice at day 3, I treated the mice at day 8, 11, and 14 with my combo. And as you see here, the tumors all grow. So there's no protection. So I made my good therapy now insufficient. And then the key experiment here that we and many others did, you take the mouse, right, naive mouse, at day -4, you give 200, 300 micrograms of the anti-CD25. You wait 4 days, you take a little bit of blood, and you see, did the Tregs go away? They go away. Systemically, they disappear. And then you challenge with the tumor. And then you put your suboptimal therapy. And when we did that, wow, great responses. Yeah, lots of complete responses and disease stabilization for a long, long time. And that's what made everybody super excited. This is prophylaxis, right? That is really not the experiment you wanted to do. You wanted to do a therapeutic experiment. So with that in mind, we basically had mice that were treated at day 0, were injected at day 0 with the tumor, and then at day +4, receive one dose of the anti-CD25 antibody, and then their suboptimal therapy. When we did this, and when you look at the papers from anyone in the field that did a therapeutic intervention with anti-CD25, it never worked. It never worked. And what you will find is lots of papers writing this: depletion of CD25 positive activated effector cells in the tumor. That's probably what's limiting the activity. This is a bad target because CD25 gets activated in the effector cells in the tumor, and then you're going with this antibody and you're killing them. What you will not find is data supporting that statement. So the statement makes sense, but there is no data supporting the statement. And in fact, all the data that I just showed you suggests that that's not the case. So Fred in the lab said, you know what? I'm just going to do the experiment. Let's see whether PC61 is depleting my effectors, my activated effector cells. Simple experiment. So mice get challenged with tumors, and then they get killed. They get treated or not with the anti-CD25, and then we look at the tumors, FOXP3 versus CD25. Untreated animals, 44% of the CD4 cells are FOXP3, so large fraction, that's known. You treat with the anti-CD25, and yes, most of the CD25 cells are gone, but they're not gone. There's still 44% of FOXP3 positive cells. And the effector cells are not eliminated. So the antibody is not failing. That was our hypothesis, because it's depleting activated effectors. It's failing because it's just not depleting the Tregs. And what we learned later on is that this antibody is engaging with CD25 in the tumor and inducing downregulation of the receptor. So it gives you this false impression that the Tregs are being depleted, but they are all there and they're quite functional. So that's when we start following the data and try to learn about things that we didn't want to learn at the time, which is Fc receptor biology. And so happened, and this is why I mentioned P61 being a rat IgG1. So happened that for an antibody to exert good ADCC or ADCP, it needs to have very good binding affinity to the activating Fc receptor 3. It needs to have low affinity to the inhibitory Fc receptor 2b. And it also needs, in tissues, especially in tissues, to have a good binding affinity for Fc gamma receptor 4. PC61 or any rat IgG1, they bind okay to 3. They bind too strongly to CD32B. That's a very strong binding pattern. And they don't bind at all to Fc gamma receptor 4. They say, well, this might be the problem. The antibody that we chose, for whatever reason, depletes in the periphery, and that reason is that in the periphery, you don't need FC4 and you can just deal with 3, but in the tumor, it doesn't. Again, an hypothesis, testable. You can take the P61 clone, resolve the variable region, graft it into a mouse IgG2A, that is the classical depleter, and recover your FC4 binding. And when you do that, you go from no depletion of Tregs, just receptor downregulation, to a 4-fold depletion of the regulatory cells. So now he fixed the antibody. So now Fred has an antibody that can deplete in the tumor. So now, can this antibody confer protection on a therapeutic level? Mice challenged with tumor, MCA205. At day 5, there is a tiny speck of palpable tumor, but it's there. You give 1 dose of the 2 anti-CD25 antibodies. And because this was never going to work as a monotherapy, we did what everybody do, combine it with anti-PD-1. This model is PD-1 resistant, so PD-1 monotherapy doesn't work. This is the data. So mice that are left untreated, they all grow tumors. Mice that are treated with the anti-CD25 that deplete Tregs in the periphery, i.e. blood and spleen and lymph node, but not in the tumor, fails. It doesn't, all the mice die. Anti-PD-1 gives you nothing, and nothing plus nothing in this case gives you nothing, right? So the combination of anti-PD-1 with this anti-CD25, with the old anti-CD25, the P61 clone doesn't give you anything. One shot of the new anti-CD25 that now can engage with Fc receptors gives you two complete responses. You see, the tumors grow pretty big, almost 1 centimeter, and then they crash. But only 2 out of 3. You combine it with anti-PD-1, and you get all the time 70 to 80% complete responses. All the mice grow tumor, and then they collapse. And that's when we got super excited about this, right? So we fixed the antibody, we learned some new biology. This is relevant to all the work that we tend to do in mice using rat or hamster antibodies, where the isotypes of those antibodies might not do exactly what you want. And this, in a way, revalidates CD25 as a target. But we still had this question, this problem. There were 2 antibodies tested in the clinic to CD25 that did not work, basiliximab and daclizumab. They did not work against solid tumors. So we started thinking a lot of this, about this, and we realized that, yes, daclizumab and basiliximab were tested, I think it was against breast cancer in humans. They didn't show much data on depletion, but definitely they didn't show any data in activity in terms of patient survival or progression-free survival. These antibodies were not developed to kill regulatory cells. They were developed to bind to CD25 and prevent IL-2 binding to the IL-2 receptor, because they wanted to use this antibody to shut down graft versus host disease, inflammation, or transplantation. They wanted to shut down effector T cell activation. Right. And what's very important is if you think about this model, and out of complete wedlock, the PC61 clone that we've used, the one that we used to re-engineer the antibody, it's a partial blocker of IL-2 receptor interaction. It's not a full blocker like daclizumab and basiliximab block 100%. These guys block 100% of phospho-STAT5. The PC61 blocks about 50%. And then we started thinking, well, if you have a tumor and you have regulatory cells and effectors, the regs express a lot of CD25 in the surface, hence they get decorated nicely by P61. The effector cells express much lower, as I show you, so less decorated. And that decoration, as I said, allows receptor cross-linking on the innate compartment and elimination of the Tregs. And that doesn't happen here because you have very little antibody. What happens when you kill Tregs? You have more IL-2 available. And if your antibody now it's binding to IL-2 receptor, not allowing the IL-2 to signal, you are wasting a really good resource. So it might not be the reason for full failure, but you're wasting a good reason. Now, in the case of daclizumab, there are 100% blockers, and IL-2 we know it's an essential factor for effector T cells. So we came out with an hypothesis. Let's make a different antibody that binds to a separate epitope of CD25 that doesn't interfere with IL-2 signaling. And that antibody should be Fc receptor enhanced, so it should deplete the regulatory cells, and then that wonderful IL-2 should signal onto the effectors with no problem and get you full immune activation. Again, best hypotheses are the ones that you can test. We made the antibody, and this is Isabel's PhD on one slide. Untreated animals, they grow tumors. If you treat the animals with 1 dose of the PC61, that is a depleter, the one that Fred made, you have 10% complete responses. So 1 mouse rejected a tumor, another one very delayed. This is the antibody that requires combination with anti-PD-1 to work well, right? Isabel's antibody, which we call anti-CD25 NIB, standing for non-IL-2 blocker, 100% complete responses. A single dose of the antibody, monotherapy, no need for anti-PD-1. This antibody, every time you work in an inflamed tumor microenvironment, this antibody just blasts away the tumors. The key experiment is the IL-2 blocking the culprit, right? Can we really demonstrate that? And very cleverly, Isabel did the following experiments. Mice left untreated, they all grow tumor. If you treat them with the anti-CD25 NIB, this next generation antibody we make that doesn't block IL-2 signaling, you have very nice tumor control in all the mice. You do that same experiment with the anti-CD25 NIP, but you neutralize IL-2 with antibodies, and you lose tumor control. That just says IL-2 is important. We know that. But the key one is that she used the anti-CD25 NIP, and she mixed it with PC61 and treated the mice with that mix. So the NIP should be doing all the killing, not blocking IL-2, and the P61 should be doing killing, but it blocks IL-2. And that's a yellow line. You completely lose the control. And that demonstrates that the problem with PC61, the reason that it doesn't act as a monotherapy, is the IL-2 blockade. And most likely, that is the problem, or the first big problem, that daclizumab and basiliximab had. So with this in mind, started collaboration with a company, and then that company got acquired by Roche, and we started an antibody campaign, and we made an anti-human CD25, RG692, 6292. And this is just testing the antibody in vitro in a culture of activation of PBMCs, and you see dose-dependent depletion of Tregs, this percentage lysis, and no dose depletion of activated effector cells, but you start seeing obviously some degree of lysis in vitro at the very high concentrations of the antibody. Most important, if you take human lung cancer single cell suspensions, you see that if you add the antibody and you count regulatory T cells in the tumor suspension, you see very nice depletion. And if you make if you make the clinical antibody now Fc silent through mutations, you lose that depletion. So this is not blockade of CD25, is ADCC. And we see the same in other tumor samples. Roche went ahead and started a clinical trial with this drug as a monotherapy first, and then an expansion towards phase 3, combining it with atezo. One of the most important things out of these trials is the toxicity profile. So a very well tolerated antibody, where the dose limiting toxicity at very high amount was a skin rash. There was no gut tox. There was not cardiomyopathies. There were none of the classical toxicities associated with anti-CTLA-4 were found on the anti-CD25, which is already a little bit puzzling if both are affecting the Tregs. But we can discuss that later. Very important, right? So translation work on those samples. What the Roche team showed clearly is that very quickly after infusion, immediately at the end of infusion, at the highest concentration, so 35, 70, and 100 mg, the patients, all of the patients, had a significant depletion of circulating regulatory T cells. This is right after the infusion. This is 72 hours after the infusion. This is 14 days after the infusion. You see that 35, 70, and 100 mg gives you very nice depletion, right? So the antibody depletes systemically and does not give you that tox that anti-CTLA-4 gives you. So that, again, it's important to consider. This is where it gets tricky. So as I've been telling you, the most important thing for a Treg depleting antibody is to deplete in the tumor, right? That was the problem with P61, depleted systemically, but not in the tumor. And the RT6292, at the target concentrations, which were 35, 70, and 100, and take this with a grain of salt, because the numbers of patients that they could do pre-post tumor biopsies to look at Tregs is very small. They never saw, they failed to see consistently more than twofold depletion of Tregs, right? So this is the twofold area. So a very low dose, you see nothing. This patient, impossible to conclude. 3 patients now at 20 mg, 1, no depletion, another one very close to no depletion, and 1 doing a bit more above. 70, you have half of the patients under 50% depletion and half above. Not impressive depleting pattern. So dose-dependent depletion in the blood, tenfold, very powerful. Low, most under twofold, and variable levels of tumor depletion. If you want this thing to work, you want high depletion and not variable. So what can explain this low and variable intratumoral depletion, which I think is most likely the cause for the antibody failing? Well, hold that thought. So in parallel, right? So when you're doing this type of research, in parallel, you carry on. And one of the things we wanted to know is exactly what microenvironment. So are all tumor microenvironments permissive of Treg depletion? Can a good Treg depleting antibody work in any microenvironment, or would depletion be heterogeneous? And one of the tumor microenvironments we wanted to study is glioblastoma. So Felipe in the lab, a very talented postdoc, and now it's an assistant professor at Oxford, he said, look, I have 3 questions. If you're going to make me work in brain and CD25, let's frame 3 questions. One, is there a role for Tregs in glioblastoma? Two, is there a machinery for engagement of Fc receptors? Are there Fc receptors, NKs or macrophages or microglia that can promote Treg depletion? And third, which I think is the coolest question, is what happens if you manage to deplete Tregs? What are the consequences? Not only tumor rejection, but what changes, right? With the anti-CTLA-4, we have this hint of data that suggests that Fc receptor engagement induces macrophage myeloid cell remodeling. So will the same thing happen with a good anti-CD25 in the brain? Is this a good approach to activate the innate compartment? Question. So he made a signature out of single-cell RNA-seq from patients with glioblastoma, and he tried in different cohorts. TCGA, there was a slight correlation on high fraction of Tregs in the tumors and overall survival. In the Chinese glioma atlas, that signature was very strong, and what was stronger on both, it was the 1-year progression. It was much higher. The events of people progressing before 1 year were higher if the patients had a high fraction of Tregs in their brains. So data suggesting that having that Tregs are bad in the context of brain cancer. Now, is the target there, right? Is CD25 expressed, and is it sufficiently expressed and selectively expressed? And it is. This is flow cytometry data from human TILs from GBM, FOXP3 versus CD25. You see very nice co-staining of FOX and CD25. So the regs express CD25. This is the histogram showing you the regs and the CD8 compartment, so preferentially expressed on the regulatory cells and not much on the CD8. Yes, there is a fraction of CD4s that expresses it. So in humans, it seems that Tregs might play a role, so we should attempt to target them, and they express the target. So let's go back to mouse. So been getting into GBM slowly but surely with my team, and Steve Pollard at Edinburgh has been a great collaborator. He's an expert on cancer stem cells, and he created several models for orthotopic transplantation of brain tumors. And we've been helping him characterize the immune compartment on those. So one of the models that he uses, and now we use quite a bit, is a NF1 knockout, PTEN knockout, EGFR positive tumor, transplantable GBM tumor cell line. Those are the only mutations. It doesn't have all the mutations that a chemically induced tumor like GL261 would have. And in this paper, we published that it's quite immune evasive and very heavy on presence of macrophages and obviously microglia, which mirrors GBM quite well. So Felipe did the classical experiment, challenged the mice with tumors now in the brain, waited 14 days, right? So waited quite long. These mice, around day 24, they start dying, right? So we're doing at day 14, so quite late. One shot of the anti-CD25 nib, and you see already a significant depletion, 4-fold depletion. That's what we always see when we have activity, about 4-fold depletion. And this is the quantification. The antibody depletes. Era of single cell genomics and transcriptomics. We run the T cell compartment, and this is the single cell map for the CD8 compartment in this mouse model of glioblastoma. And nothing new. You have all the usual suspects from CD8 naive-like to activated, proliferating, exhausted progenitors and progenitor exhausted. What's nicer when you do the single cell, and we do always, is just run TCR-seq as well. And that allows you to look at clonal expansion. And now we start seeing very interesting differences, primarily in the clusters of proliferating CD8s and effector CD8 T cells. Those are the ones that we find hyperexpansion of TCR, suggesting that there is antigen engagement and T cell expansion, and concomitant with the quantification of T cells that we do by flow cytometry. This is the quantification. So basically, mice left untreated, mice treated with the anti-CD25 nib, they show a significant increase in granzyme B expressing CD8, also a significant increase in PD-1 positive, highly proliferative CD8 T cells in the brain. If you neutralize IL-2 with an antibody, you lose the expansion of those populations. And again, this is consistent with what we demonstrated before, so nothing too new. Classical experiment, obviously, do these changes lead to tumor control? in this very aggressive tumor? And it does. 50% of the mice get cured, and we kill them after 150 days. But this is quite amazing for this type of tumor and consistent with the role of IL-2. If you neutralize IL-2 or if you deplete the CD8, you have a significant loss in tumor protection. So again, nothing super new and exciting, but it makes sense, except that we have really good anti-tumor activity. One of the other things you can do with the transcriptome and with TCR-seq is to look at clonal relationship, right? So what population is differentiated into what population? And you do that by looking at each cluster and then looking at the extent of TCR overlap that it exists. And not surprisingly, there is quite a bit of overlap between effector T cells, the proliferating T cells primarily. Where you don't see a lot of overlap, and I find this quite interesting, is population C6 and C5, which are the exhausted CD8 cells. They don't share many TCRs with the effectors and the proliferating CD8s, suggesting that it's not that we are reactivating exhausted cells, right? They are left to their thing. This is suggesting that it's new sets of cells being primed and activated, and those are my effectors and proliferating. That's what the data suggests. And the fact that the exhausted cells were not changing much, we said, well, maybe this will be a good reasoning to combine with anti-PD-1. PD-1 impacting the effectors and the anti-CD25 creating, PD-1 impacting the exhausted, and the anti-CD25 creating better effectors. And that's pretty much what happened. So the anti-CD25, again, very nice protection. The anti-PD-1, about 20% protection, and the combination gives you significant improvement in survival. Now, where I wanted to get, sorry, it took me so long to get here, is beyond T cells, right? I talked to you guys about it a little bit about Fc receptors and innate cells and how important they can be. So we wanted to look at them in the context of this Treg depletion. And again, the brain microenvironment is fascinating in terms of the diversity of the resident innate compartment or microglia, and then the inflammatory infiltrating compartment. And we do high-dimensional FACS when Felipe does that, and then look at differentially expressed, differentially abundance of different populations. He sees, when looking at the innate compartment, significant changes. So everything above this line is significant, and this is what increased with anti-CD25. Primarily, there is a cluster of CD8 cells, monocytes, that seem like they're just getting into the brain because they don't look very activated yet. And this population, C1, that is the one that I'm going to focus, which are monocyte-derived macrophages. They are highly activated, and they express very, very high levels of Fc receptors. And we thought, oh, that's interesting. So now we have macrophages that seem activated, and they also express a lot of Fc receptors. And they also had there is a transcriptional signature called FAT, PHAT. They express high level of that signature. So they're highly phagocytic in response to the CD25. So this is the quantification of those cells, so a significant increase in that Fc receptor high MDMs. Important, when you block IL-2, that population decreases, right? Suggesting that somehow IL-2 plays a role there on the presence of this population. But more logically, when you neutralize interferon gamma, you completely lose that population, right? Suggesting, and we know that gamma can have an impact on the CD8 compartment. Long story short, we did some spatial transcriptomics, and what happens is that the IL-2 is essential for the CD8 compartment to get activated, and we knew that. Then they produce the gamma, and the gamma is essential for the macrophages to change. But who cares, right? They're changing. This could be just a consequence. It could be completely relevant, just a description. And it could be completely relevant if you ignore it. But because we're so much now into understanding these interactions between antibody, target, and an effector innate cell, we thought, well, if you have more Fc receptor on your innate compartment, right, you have now turbo boost them to make more depletion. I don't want to make more depletion of Tregs because they're already being depleted. But you could try to target the tumor, right? This might be a window of opportunity where you turbo boost your innate compartment to then deplete or kill a tumor or eat a tumor cell in presence of the right tumor targeting antibody, right? In that way, you eliminate the tumor. So there is potential increase in ADCC, ADCP capacity. And we saw that on the FAT signatures and on the expression of Fc receptors. And again, that's testable. We were working with Martin Pule.
你从上面，你从上自己站定，对。
Sorry. So we were working with Martin Pule that was developing CAR T cells against EGFR V3, and he gave us the sequences of his single chain FVs, and we took that information to make our own anti-EGFR V3 antibodies. EGFR V3 is a common target in human glioblastoma, and it's expressed in our mouse model. Right, so we basically did the same protocol. The mice received either anti-CD25, a single dose, and then we, because this was never going to work, we just like bathed the animals in the anti-EGFRB3, which is also a mouse IgG2A, so a good depleter. And this is the data. So if mice left untreated, they all die a few days after day 40. If you treat just with the anti-EGFRB3 with the tumor targeting antibody, it's a nice antibody and it's quite high affinity for Fc receptors. So it does a pretty well. It doesn't do a good job, but it gives you 20% complete responses. So that's not horrible for this model. As I mentioned, the anti-CD25 monotherapy, 50% complete responses, but the combination of EGFR, anti-EGFR, and anti-CD25 gives us now 100% complete responses in this model. So impressive anti-tumor immunity in terms of functional, flow, single cell, but also at the level of tumor protection in this combination. What's very important more mechanistically is that when we take the mice treated with the combo treatment, but we add to that the anti-interferon gamma, the one that blocks macrophage activation, not T cell activation, but macrophage activation, we go back to the 50% complete responses level. So we go back to just using the anti-CD25. And I hope that's clear, right? So what we're unveiling here, and this is what we think is the mechanism, but I'll describe already, what we think we're unveiling is, yes, you can deplete Treg, and they act the way we know it acts, activate the CD8 cells, and CD8 proliferate more, and kill more, and wonderful. And that gives you a level of anti-tumor immunity. But it also gives you an opportunity because it's turbocharging the max. Now, if you don't take advantage of that opportunity, you remain here at 50. You need to take advantage of that by adding a rational combinator, and that's anti-EGFR or any tumor targeting antibody, and we have validated this. And if you eliminate now the turbocharging capacity by neutralizing gamma, you go back to the levels of just anti-CD25 protection. So that's all mouse, right? So in the last 2 slides, that's all mouse. So what happens in humans? So we've been working with human tumor fragments in the lab from different type of cancers, but this is data now done exclusively with GBM. Patient with GBM goes into the operation room, the tumor is removed, a piece of the tumor comes to my lab where the team dissect the tumor and make little fragments, 2-3 millimeter fragments. And those fragments are put in vitro in special media. And you can use this as a perturbation experiment or proof of mechanism experiment. These fragments will be alive, i.e. the immune cells will be retained and functionality will be retained for about 24, 48 hours maximum. At least that's the current protocol that we have. So you can perturbate. You can, let's say in this case, add a Treg depleting antibody, the one that was in the clinic, and test whether it does what the mouse antibody does in mice. So again, a proof of mechanism. It's a model. All models are flawed. But this is one way to get closer to understanding how this antibody should perform in a human tumor microenvironment, right? That has been excised. And this is the data. So this is a fresh tumor sample from brain cancer, human, again, all human, FOXP3 CD25, very nice infiltration of Tregs. When you leave these fragments untreated overnight or treated with control immunoglobulin, there is no changes on the Treg population. And when you add the anti-CD25 NAB overnight, you see very nice, well, you see depletion of regulatory T cells. So the antibody can do it. And in fact, it does it in the vast majority, not all, and this is important, of the experiments we tested. There is a significant, each one of these is a pair experiment on a tumor from different patients. So this is 6 different patients that we did the experiment. There is depletion of Tregs, and consistent with the depletion, we see also that the CD8s increase the levels of granzyme and Ki67 expression. So basically validating the mechanism that we observe in mice, in a mouse brain tumor microenvironment, we're observing it now in a piece of fragment of a human brain tumor. What's very important here is the extent of depletion and the heterogeneity on depletion. This is very heterogeneous, and we see some tumors actually that we see no depletion whatsoever. And that's not a problem, because that's exactly what we're trying to solve. What this data made us realize, which it will sound obvious, but you need to address it, is that obviously the landscape in the tumor, in a single tumor of a patient, will be quite heterogeneous. And by landscape, I mean not Tregs and CD25, but innate effector cells and Fc receptor expression. So we think that this extent of heterogeneity is at the cellular level and molecular level dictated by the activity and extent of presence of the right macrophage subset. And that's where we're taking this research to try to understand that. And in fact, not only that, but can we perturbate the system further to ascertain activation of the right macrophages? And this is just preliminary data done incubating these tumor fragments with interferon gamma because of the data that I show you. And then this population, which is MDM CD49 high, this is the equivalent of the Fc receptor highs, and they express the highest level of Fc receptor. That population significantly increases when you treat the human GBM explant with interferon gamma, right? Kind of offering a potential avenue, right? Maybe not necessarily with gamma, but at least a line of thought of activation of the innate compartment to allow proper depletion of regulatory cells and proper activity of tumor targeting antibodies as well. So with that, I leave you with my thoughts. Multiple targets for Treg depletion. You guys probably know, right? So CTLA-4, CD25, and CCR8 have been the ones that have been explored the most, I would say. And in my book, they're all good targets. They have their pluses and minuses, but they are quite good targets. And then a bunch of other ones that I would say they're horrible targets for Treg depletion for many reasons. But the primary is that either they don't deplete or they deplete effector cells as a bystander effect, and you don't want that. So what should we do as a lab? All right, this is my dilemma, but we resolved it. Should we make another? Should we keep on doing single cell? Should we look for new targets for Treg depletion? Do we need more? We have 3 that are quite decent. They have their issues, but they're quite decent. Or instead of focusing here on the target, should we be instead looking at this part of the tumor microenvironment? Should we be looking more at the innate compartment and the functional ability, not doing atlases, but perturbating the innate compartment in human tumors, right? And understand how those cells function or fail to function. And this is just a snapshot, just to show you the challenge. This is expression of one of the 5 Fc receptors in human GBM. And look at the level. So this population bimodal, this express a lot. This doesn't express nothing. This express maybe a little. And this is just the quantification of expression, percentage of cells that express this particular Fc receptor in granulocytes, NKs, intermediate monocyte, blah, 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 blah. It's super heterogeneous. And obviously not because this is heterogeneous, this is going to be difficult. But those are the questions we think we should be spending the time, right? So should we focus on understanding and modulating this innate TME, right? Could I find a drug? Would gamma get everybody just aligned, boom, up for Fc receptors? Or is there something else that could do it, right? So that's what we want to be doing. We want to embrace this complexity. And we want to transition also at the side of antibody engineering from indirect targeting of Fc. And by that, I mean, the antibody will bind to my CD25, and then the Fc portion will bind to the multiple Fc receptors. So that's indirect. Or direct, should I be targeting with Fabs or different types of antibodies directly the specific Fc receptor in the specific effector innate cell? That's the question that we think is the important one. So with that, I hope I convinced you that Tregs are an important target in GBM. The CD25 NIP is a very interesting tool in the fight against cancer, but it has its limitations. The human one has limitations. We believe that those limitations are primarily based on its incapacity to systematically promote very good depletion in the tumor, right? the mouse data, I would say it's validated, at least part of it, on the human, and create an opportunity, right? So the data on the combination of anti-CD25 and anti-GFRB3, it's promising for a mouse model, but also there is plenty of room for improvement, especially translating this into humans. And I think the big challenges that we're taking in the lab is, one, can we systematically enhance Fc receptor activity within these variable and plastic TMEs in order to maximize the activity of ADCC or ADCP antibodies? Or should we be targeting or should we be developing therapeutics that directly and specifically activate the macrophages through engagement of particular mixes of Fc receptors? With that, I just want to thank our founders, Cancer Research UK, primarily additional ones, the Cancer Institute, my amazing team of basic and clinical scientists that are really tremendous. In red, the people that did the work, that my alumni. This has been researched through many years, and then the guys in the lab now pushing this forward. At Roche, Maria Mann, which was our key collaborator. At Tusk and Guvier, our key collaborator, they were the ones that believed on our concept of the new anti-CD25. And then our wonderful colleagues on the clinical side, Tracer X, the Swanton team, Litchfield, Kerry Chester, and Martin Pule. Thank you very much.